then please turn off your camera. So it's a great pleasure to invite Adam Kleczkowski from the University of Strathclyde to come and present here. Adam initially completed his education in Poland before doing a short postdoc in Germany. Then in 1992, he moved to Cambridge University, where he was for 15 years before he came to Scotland, where he was at 10 years at Stirling University. And then 5 years ago, Adam joined Strathclyde. I believe he collaborates with one or two of us here at the Institute, so it's not entirely unknown to us. And Adam is going to talk today about sort of his work on modelling plant epidemiology, and particularly around trying to understand and introduce variability into his model. So without any further ado, Adam, I'll hand over to you.
Yeah, thank you very much, Rob. And it's my great pleasure to meet everybody here in the, well, not in person, but online. And I'll be talking about taming the variability in plant pest and disease outbreaks. And just as Rob introduced the talk, there will be a bit of modeling there, and there will be 3 slides with equations, and I apologize for those in advance, but hopefully you can bear with me over those 3 places in the talk, but hopefully there will be enough non-equation, non-mathematical stuff to interest the general audience as well. Let me just get the... So the plan of the talk is, after a brief introduction, I'm going to talk about giving you the idea of why we're interested in looking at the variability. I will be then talking about 3 examples, and 2 of those are on small-scale experiments, particularly looking at biocontrol, efficacy of biocontrol of soil-borne diseases. And the first example will be addressing the question, why do we need variability? Why do we need to talk about variability? Example 2, again, talking about Rhizoctonia celani and radish system, using variability to estimate risk, and particularly looking at variability that's hidden from our normal view. In example 3, I'll be moving up the scale and bringing in field experiments, so moving to more a larger scale and longer periods of time. And I'll be looking at an example from Ash dieback. This is work that is ongoing. And the question here is looking at finding resistant tolerant trees, or identifying phenotypic variability in the ash tree population with a view to combat ash dieback. And then I'll finish with essentially a 4th example where I will be looking into the future. So talking more about the projects that I'm slowly moving into, and looking at high throughput assessment of essentially phenotypic resistance to disease, but I'll be also looking at modeling growth, plant growth. And then finish with a bit of a summary and hopefully wrapping up the discussion of the variability. I just want to start by saying that variability is part of every biological system, and it spans the scales from genes to and cells to populations and the whole continents. Now, very often both in science as well as in practice, variability is treated as a nuisance, or something that we're striving to avoid or reduce. It's also very often thought of as a risk. It's associated, I will be talking about this in my 2nd example, but it's associated with a risk of something bad happening. And again, our actions and our thoughts are about trying to reduce the variability. However, I want to convince you through my examples that studying variability can actually give us an unprecedented insight into the dynamics of natural systems, looking at how natural systems behave. both natural systems and systems and agricultural and experimental systems. And I want to argue here that variability is both a problem, something that we clearly want to reduce in many cases, but it's also a solution. So it's something that we might be wanting to introduce into the system because it helps us in achieving our goals. Two quotes here. The first one from William Osler, who was a Canadian physician and one of the founders of the Johns Hopkins Hospital, which became very, it's a famous hospital in the US, but it became very famous over the last 3 years when it was publishing the COVID data. And the quote is that variability is the law of life, and no two bodies are alike, and no two individuals react alike and behave alike under the abnormal conditions which we know as disease. And obviously, William Osler is talking here about human diseases, but the same thing does apply not only to pests and diseases of plants, but also to their growth patterns and their response to the environment. And then more recent quote from Nassim Nicholas Taleb, who's famous for author of the book of The Black Swan, introducing the concept of a black swan. And that's that the central illusion in life is that randomness is a risk and that it is a bad thing. And that sort of goes back to what I want to convince you, that variability is both a problem and an interesting thing to look at. Now, I just a bit of a trip down the memory lane. I actually was at, as a relatively young postdoc, was at what was then SCRI in Scotland back in 1994. And my host, John Crawford, who was at SCRI at the time, and some of you probably remember John, who then moved on to Abertay, Sydney, Rothamsted, and he's currently at Glasgow. I remember when I visited SCRI, John took me for a walk around the campus, and then we stopped at one of the field trials and he showed me 2 plants that were growing in that trial. Unfortunately, after almost 30 years, I don't remember what plant we were talking about. But anyway, he pointed to those 2 plants and said, well, it's a big question of why those 2 plants are different. Why they, I mean, they were very similar to me, particularly as a young postdoc just moving into the field of plants and plant diseases. But it was clear that they are different. There is something different about them. And there are different sources of variability, there are different sources, there are different reasons why the 2 plants might actually be different. And there could be a variety, there could be different varieties, there could be different, they could have different genetics, and they could have different phenotypic characteristics. They might be growing in a different microenvironment. So even if they are growing very close together, they might actually be getting different, slightly different soil conditions, slightly different water availability, maybe exposure to wind. And that would cause those 2 plants to look differently and also to respond to pests and diseases in a different way. If we look at the disease spread, then spatial component, particularly location related to source of inoculum, will be a big factor in deciding on how the plant responds to the disease and to the pest. And there are also many other underlying processes that we might not be able to characterize or give them a name. And in mathematics or in modeling, we usually call them stochastic elements. So it's essentially the toss of a coin or a throw of a dice that determines the response or the current response of a plant. Now, I want to also argue that plant pathogen systems are a really good case for studying epidemiology in general, and particularly looking at the variability in epidemiology. They're easy to replicate, and they're easy, therefore, to study variability. They're relatively easy to have treatment and loss, and there is usually no ethical consideration, unlike working with humans or animals. And also, as we know, they're fundamental to food chain. So I want to, in this introduction part, I just want to briefly introduce the modeling concepts, or the concepts that underlie the epidemiology, plant epidemiology, starting with the disease triangle that was introduced back in 1950s, that encompasses, that looks at the interactions between a pest and a host, and also incorporates these different factors associated with the environment that influence both pest and host behavior, and also influence how they interact. What we're going to look at from the modeling perspective is really this arrow between the pest and the host, is the way in which the pest interacts with the host and causes the host to become infested or infected. And I will be using here the so-called susceptible, infected, recovered, removed paradigm that again became, a lot of people became familiar with, as it is the same paradigm that has been underlying COVID modeling over the last 3 years. And so the plants under this paradigm, the plants are initially described as susceptible or healthy, then either through primary infection or infestation that originates from inoculum, that could be soil-borne inoculum or airborne inoculum or waterborne inoculum, the initial plants become infected. And then the infected plants might again produce the further inoculum and cause the secondary infection. So we have this cycle that keeps producing more and more infected plants from the healthy ones. And then the plants can, sometimes can recover, or very often the plants die. And that's the process here is also included in that model. So it's called an SIR modeling, and because it goes from susceptible to infected to recovered, where we call S, I, and R, by those letters, we call those different elements in the population. So that's really looking at how pests and hosts interact. But then we know that there are different sources of variability, some coming from environment, as I was talking about, differences in soil and water contents. The weather is often a very important factor, the changes in temperature or humidity or rainfall. And in all longer terms, we have the climate change that again will affect different parts of the country or the globe in different ways. Host can have different sources of variability due to phenotype, location, age, and then again pest, different genes, different varieties of a pest, different developmental stages, and potentially competition with other organisms. So what I will be doing in the next 3 examples, going through this next 3 examples, is looking at how can we identify these processes, these elements, the modeling elements in those different systems, biological systems. How can we then build in variability and the dynamics of the variability, and how the studying of the variability can help us in understanding the processes better. And one of the things that I want you to think about when I'm looking through those examples is the importance of variability, but also the importance of how we approach the studying of variability, and particularly that the fact that that might need us to rethink some of the paradigms that underlie our experimentation, particularly looking at highly controlled, highly replicated systems and frequently observed systems. So the first example, that's an example that we studied when I was first a postdoc and then a lecturer in Cambridge throughout 1990s and early 2000s. And this is a system that Chris Gilligan in Cambridge and others and his collaborators, including myself, we've been developing this system. And that's looking at an important soil-borne pathogen, Rhizoctonia solanae, that among other things, causes dumping of seedlings, and it attacks many different species. The system that I will be showcasing here uses radish as a host, not necessarily because radish is a crop of the future, but because it's a nice experimental system that's easy to use and very quick. And that's because we wanted to keep the environmental and phenotypic variation under control. These were the radish host and the pathogen Rhizoctonia solani were both grown in a sand medium, a sterilized sand medium. Again, I'll show you the examples of this experimental system in next slide. We also looked at we're particularly interested in how the fungus spreads, how the epidemic, small-scale epidemic follows through time, but we're also interested in the impact that biocontrol agent, in this case Trichoderma viridi, and other Trichoderma species were used in that context. And this experimental system, I want to stress here, I'm using that as an example of a highly replicated, very quick system where the variation is highly controlled, but what we're looking at is capturing the variability. Not going into great detail here, just to show that life cycle of Rhizoctonia solani, quite a complicated life cycle, particularly growing through mycelial growth on soil, then mycelial growth on surface of the host, and then penetration of the host through by the mycelium, and then leading to the wilting and death of infected plants. And this is the effect of Rhizoctonia solani on radish seeds. You can see this dumping of lesions, black lesions on the plant. The experimental system, which, as I'm stressing here, because we want to address variability, what we want to start with is a system where as little introduced variability as possible. So this is an example of a system including, this is 50 radish plants grown from seed in a sterilized sand, grown in a highly controlled environment. This is the spatial arrangement. So we have 50 plants, 50 radish seeds, and then germinating, and also a certain number of inoculum units being introduced into the system. And then this inoculum grows and then catches the first and then the subsequent plants, and and they're easy to score because there are very clear visible systems. So it's an experimental system that allows very high replication, but also very high level of control. Now, it was quite surprising when the experimental results came back from this experiment, was that there is actually despite trying to keep the exogenous variability very, very low, we actually observe very high level of endogenous or internal variability in the system. A graph here showing the number of plants showing symptoms among 50. 50 is the max, it was 50 plants in a box over time, and it's a very quick system. It's finished, the epidemic is essentially finished by three weeks from planting. And each of these lines here and points represent a single replicate. So there are two things here. I mean, one is we see this characteristic shape where we're starting with primary infections followed by secondary infection as the fungus spreads from a plant to a plant. The second feature is that the infection becomes frozen, and that's due to the fact this is a dumping of disease, so it affects seedlings. As seedlings mature, the fungus stops being able to invade the plants. The plants become a bit stressed afterwards, which is why we see this small pickup of the disease a bit later. And the third thing is that there is a high level of variability. So in some replicates, we see about 1 in 5 plants being infected. In some replicates, it grows up to about 60%. How can we model this? How can we describe this? So we're again breaking down this process of soil to plant to further plants, the spread of the fungus into these two components, where we're looking at the primary infection, so infection that originates from the soil inoculum and then moving to the first plant that it encounters. And then this first encountered infected plant becomes a source of inoculum for other plants, and we get essentially secondary infection. And we're capturing that, as I described in the introduction, by this susceptible infected recovered model, where we introduce those two sources of infection as well as a potential recovery of the hosts. Now, so this is the concept. I'll talk about model results in a moment. But I just want to first start with looking at why variability is important and what does it tell us about the system. So if we compare the output of the outcome of the experiment for without biological control and with biological control, we get, and we're just looking at average values over the 5 replicates in one case and the 5 replicates in another case, we see that Trichodoma viridi has reduced the level, the dumping of disease from average 25.8 out of 50 plants to about 14.8 plants, and then we're looking here at that point here. If we just wanted to compare the final outcome, I haven't shown you the results with the Trichodoma, but if we just wanted to compare the final outcome, the reduction is actually not significant. So if we look at just the values at the end of the epidemic, and we're comparing the results without and with Trichodoma, is there is this reduction in the final outcome in the number of average number of infected plants, but there's this huge variability here that makes the reduction not significant. In fact, the p-value would be 0.1478. That would have been normally an end of the story. But there is actually much more to the story when we start looking at the variability. So if we look at now we're comparing here on the left-hand side, the graph that you've already seen without biological control, single populations, 5 populations, increasing the number of infected plants, but also increasing variability. And then as we look at the case with biological control, you see this huge variability in the response here. So firstly, we get this quite tight increase for each population, but at the same time, the populations differ massively from the case where control was entirely successful. There was no apparent disease in the system, and the population where your control failed. Absolutely, I mean, it actually was even worse than in a situation without biological control. This is my first equation, and I'm not going to go through these details of this equation, just to show you that there is actually an underlying mathematics in that that gives us the credibility of approach. We've looked at the estimation of parameter estimation for this particular relatively simple model. We used Bayesian inference, so the results shown here on the right-hand side are what's called posterior distributions. In a sense, they give us an idea of what's the probability, what's the chance that were we to observe an epidemic, it would be behaving like if the parameters were of a certain type. And well, if you just look at these 2 parameters, so this system has 3 parameters, one corresponding to the primary infection, one giving us the strength of secondary infections, and one giving us an idea of how quickly plants mature and therefore stop the fungus from spreading. And if you compare those results for the case with and without the biocontrol agent, there is very little difference in those 2 parameters, but there is a big difference for primary infections. In other words, we conclude from that the biocontrol reduces primary but not secondary infections. And therefore, what happens is that it's the secondary infections that generate the variability. The primary infections essentially set the scene, and then the secondary infections, through its exponential growth, very fast growth, create this large outbreak. And that's really what you see in those graphs. The primary phase just sets the scene, and then the secondary infection magnifies the variability, and then the variability is essentially frozen by the fact that the plants mature. So concluding that, looking at both the variability and looking at how variability changed over time, and delving into our understanding of the dynamics of variability gave us an insight into the biological processes. And we also ran some experiments using different media, using different biocontrol agents, found that moving from Trichodoma viridi in sand to Trichodoma hartzianum in sand, we found that both primary and secondary infections were affected, and in soil with the added complexity, this is mostly secondary infections that are being affected. So it just varies from a system to a system, but the looking into variability gives us a much better insight into the biological processes. So just to summarize this first example, we've looked at the highly replicated and highly controlled system where we try to reduce the external variability as much as we can, just being able to identify and look at the internal variability in the system. We found that by looking at the dynamics of the variability, we found that biocontrol reduces infection, primary infection, but increases the variability. And we could then try to understand the processes that underlie this transmission. The disadvantages, firstly, it's a highly stylized system. It uses, not many systems are using growing radish in sand, but there could be applications, particularly as we're now moving into vertical farming, when a lot of these systems are designed to reduce variability as much as we can, growing into sterilized media, and trying to keep the conditions as uniform as possible. But there's also limited potential for exploring other sources of variability, and I'll be coming back to this in my third example. The second example, and I'll go a bit quicker through that example because I already have told you how the modeling works and what the key concepts in the modeling are. But I'm going to look here at the idea of a hidden and cryptic infestation. And the motivation for this study was that we were involved in a BBSRC funded project that looked at the transmission of soil-borne diseases, in particular Rhizoctonia solani, between propagation and field. And we're looking at the seedlings grown in glass houses, and then propagators who grow those seedlings in the glass houses are very worried about the transmission of the infection from glass house to the field, because the infested plant might cause large disease outbreaks. And the question was, can we design a surveillance system in which populations, the trays for growing those plants, are subject to visual screening for symptoms? But I'm using this example to showcase you how variability translates into a risk, and how understanding the levels of variability impacts the way in which we design the surveillance system. It's a slightly more complicated system because we're looking here at those at several stages in the propagation. So starting with the reservoir of infection, and it could be peat, it could be seeds, or it could be environmental factors, for example, rainwater. There is a population in transfer, which is essentially the propagation tray being grown in a glass house, and that will have both primary and secondary infestation and potentially appearance of symptoms. And then under successful detection of symptoms, we can take up control action, for example, chuck away a tray of infested plants. but the unsuccessful detection of symptoms can lead to an infection of target or susceptible population. And of course, there are these different losses associated with this. If we actually transfer the disease into the field, where we can have sort of millions of pounds being lost. A very similar experimental system, a bit bigger. Well, we used infected plants, but we used many more replicates. Again, the same system with radish and Rhizoctonia solanae, the same outlay, and then growing in sand. We've also done experiments growing in peat, but I'm not going to talk about those as they introduce another source of variability. So again, a very highly controlled environment. But again, as you see, there is a huge variability in this different response to the disease. But we were interested not only in looking at the symptomatic disease, but we're particularly looking at the cryptic disease, so the disease that is not visible. And that usually extends much further than what we actually can see. So if you look at the tray of plants here, then there will be just this area here where the plants have the show symptoms, but then there is a larger area that plants do not show symptoms or necessarily show symptoms, but where the soil-borne disease is present. And what we used a combination of visual scoring and destructive molecular techniques, particularly looking at the ELISA, to actually study where the plants are, both infected plants with the symptoms. So if you, for example, look at that replicate here, again, 10 plants, 5 inoculum units shown here as black dots. Red are plants with visual symptoms. Gray are places or regions, cells where the plants do not show symptoms, but where we detected the fungus using the molecular techniques. When we looked at the experiments, then the way the experiments were set up, it was grown in growth chambers in Cambridge. We used 25 populations, 50 plant each, but we looked at the full history of visible disease symptoms up to a sampling point. And we've used 5 sampling points where at each, the 5 populations were sampled destructively, and we've looked at both the symptoms and the cryptic disease infestation assessed by molecular methods. And that gives you an idea here. This is the 5 replicates that were the final, the longest running replicates, where this is symptomatic plants and this is the number of cells or plants that we assessed as having been infested. Again, we used a slightly more complicated model here, where we have both susceptible, infected, not showing symptoms, and then showing symptoms, so symptomatic, pre-symptomatic, and symptomatic plants. And we've looked and we were successful in describing not only the average or the behavior with the sigmoidal growth, but also describing the variability among those plants, both for the pre-symptomatic and then symptomatic cases. So pre-symptomatic here in black, and then in red, symptomatic ones. Again, we were able to estimate the parameters and get an idea of what the variability in those parameters is, and use this to predict what the population behavior would be. In other words, we can now start building the scenarios in which we can predict how many plants there will be, both symptomatic and asymptomatic, in the population at any given time. And you can see here this large variability in both cases. It also gave us an idea of looking at the relationship between cryptic infection and symptomatic infection. So looking here at different times, symptomatic plants on the horizontal axis, infested plants, so those who are both symptomatic and asymptomatic on the vertical axis here. The dots, little dots here, points, the circles represent the actual observations. And the color here shows the predictions of a model. So essentially, it tells us that we capture this relationship between the infestation and cryptic infection and symptoms quite successfully. Red means a high probability, a high chance of observing this combination of infested plants and asymptomatic plants, and blue meaning the chances are very, very small. And that also allows us to look at to do some predictions and to start understanding what the role of variability is in this in producing risk for the transmission of the disease from one's stage of propagation to another. And that is a practical application. If you look at this is a picture taken in the commercial glasshouse, where we're dealing with potentially millions of plants. These aren't radish plants, by the way, but those are brassicas. And then somewhere here, there is an infected plant. So the question is, how do we actually build up a decision system that how many plants we need to look at, how diligently we need to look at the plants, and particularly we can try to quantify this by using this threshold for detection and threshold for damage. So threshold for detection being minimum number of symptomatic individuals that can be detected, because we might not be able to detect all the plants, and then threshold for damage is the minimum number of individuals that can cause damage when transplanted to the field. We can set both to 0, which means perfect detection, and a very high sensitivity. And we can use this this concept of this distribution, this understanding of the variability to translate into the risk, and particularly this is the colored here, or shaded area is detection below which symptoms are not visible. So that's something where we do not see the symptoms. This area here, shown here in black, is where there is a damage caused in the target population, and below that is the region where there is no damage. And it's really the combination of those two factors, so the fact that we cannot detect and it causes the damage, that is of a risk. And we can use this understanding of variability to actually quantify the risk and to look at the percentage of populations of traits that cause problems. Not going into great detail here on the graph, but essentially I want to show you that there is this region here in the middle where the percentage of population or the risk is very high, regardless of how tolerant we are and what the detection threshold is. In other words, if we detect everything, then of course there isn't any percentage of population causing problems. If we detect a lot, then if we detect not enough, then there is a high potential for the damage. If we detect a lot, then there is there's actually not a very big chance of a damage. So just to summarize that part, this is a combination of observations and molecular tools that helps us in demonstrating infestation beyond visible symptoms, but the capturing of the variability, being able to address the dynamics of the variability, allows us to capture the risk of transferring infestation to the field. Again, the disadvantage of that system is, again, this is quite a stylized system requiring molecular techniques and a very careful study, both experimental techniques and very careful study of the models. The third example that I want to go through, and this is work in progress, so I'm only going to go as far as in the analysis of this data, and that's an example from a bigger scale, and it's an ash dieback example. The ash dieback, which I'm sure you have heard about, sort of 2012 was quite a lot about this in the news. It's a fungal disease that affects the tree, the iconic tree in the UK, ash, believed to be present in Eastern Europe in around 1992, in the UK, possibly from 1990, certainly from 2004-05, and first detected in 2012 when the epidemic was already in full swing. There's a very high mortality, particularly among young trees. The estimations are that almost all ash trees in the UK are infected. The older trees might be able to shed the infection to survive, but among young trees, the mortality rate is about 85%. There's an evidence of resistance, but it's a big question that is being asked, is, are there any ash trees actually resistant or tolerant to the disease, and how can we quantify this resistance? Also with the hope to link that to the genetic element. This is an experiment that we did not set up, but was set up by Forest Research in collaboration with Future Trees Trust. The ash was planted in Oxfordshire in 2003, not for dieback monitoring, and then they started monitoring in 2012. The site contains 39 provenances, and we're hoping that different provenances from different areas in Europe and UK, that some of those might be more resistant than others. 117 replicates across 3 blocks, 4,000 trees in total. This is how it looks like. This is pre-setting up, so it's the Google Earth picture from quite a long time ago. Then, so this is pre-2003. This is how it looks like when we have the epidemic, the trial there. This is the current of the most recent picture, where you see a lot of bare ground, and this bare ground actually contains a lot of dead trees that don't remove the dead trees. This is the disease severity over time from 2016, 17, 18, 19, 20, 21. We do have data for 22 as well. Green is essentially healthy or almost healthy tree. Red is a dead tree here. So you can see this progression, but also you see variability. And there is both variability due to the block. So there is an environmental variability, but there's clearly a variability due to the different provenances. We've used a more complicated model, and you will be pleased to hear this is the last equation I'm showing in the talk. We looked at a bit more complicated model, including susceptible different stages of infected and dead trees, with uncertainty captured by varying parameters. And just to show you that the model actually does work and captures not only the average behaviors, but more importantly, captures the variability among those different blocks. So this is different blocks, and this is different, just examples of different provenances. So hopefully different response of the plants. We've looked at particularly two parameters. The first one looking at how fast the disease spreads, and the second one looking at how fast the plants respond to the disease. And some analysis, this is very early stages of the analysis, show that there is a significant effect of the environment. So there is an environmental factors affecting the response, but there's also a very strong a lot of difference between a group of provenances and the rest. A very typical graph with the multiple gene resistance, and then some clustering. So some of those provenances, some of the trees from provenances, possibly disease spreading slower. And there is, and that's the second one, is a parameter which tells us about how long until the death of the tree. And again, we see a lot of variability, but potentially affected mostly by the environmental factor rather than any differences between the different plants. We did some, so as I said, this is a work in progress, but we did some analysis, earlier analysis, just looking at the data, and we potentially are finding some clustering in location with the trees coming from provenances more to the north are being more resistant or more tolerant to the disease than those in a sort of middle of Europe, with another cluster here at the bottom. So just to summarize that, as I said, this is work in progress. We have been able to successfully capture the disease progress in those different trees, but we also were able to capture the variability. So we're using the models now to partition the variability into the environmental component, but also the variability among provenances and variability within provenances. And the next question will be which provenances are different to the rest and which are not, and possibly hopefully will give us an idea of how to classify them into susceptible, resistant, and tolerant. So it's moving into the understanding of what the role of genetic or phenotypic variability is. And I want to look at the future. So this is the work that is, well, very much in progress. It's actually only just starting. And that's working together with colleagues in the International Crop Research Institute for Semi-Arid Tropics, ICRISAT, in India. And the reason why I'm so interested in working with colleagues there is that I'm looking for an ideal system that can be used to understand the variability. And that should have a controlled environment, but being able to introduce other sources of variability in a controlled way, a realistic setting, high replication, and possibly automated scoring. So rather than a colleague of mine back in Cambridge looking at each individual plant, we can actually do this in an automatic way. ICRISAT has fantastic facilities. They operate a field-level dual scan plant eye, which is the facility that is essentially a set of cameras that are mounted on a big crane and then can scan 4,800 microplots in less than 2 hours. So they can actually provide very high resolution both in time and space of the parameters like canopy dynamics, growth rate, early vigor, transpiration, and others. And that's automated big data analysis. So it's an ideal system where we can start looking at the variability in different aspects, including spread of diseases. We've already started looking at some of the data produced by ICRISAT. This is the leaf area size for 384 cultivars of sorghum replicated, so 4 replicates of each of 384 cultivars, and 285, so there's lots of data time points that we're converting to thermal time, and then we're doing this on a daily basis, and then my MSc student used a logistic function to actually fit to those data. The analysis is very early, but I think it shows the potential here for doing experiments where we're actually looking at really large numbers. So I mean, it's being able to, in a controlled environment, to scan 384 cultivars with 4 replicates and with a very high temporal resolution. We can link that in ICRISAT with Center for Excellence on Climate Change Research for Plant Protection, which has the temperature and CO2 enrichment facilities. And one of the things that we're planning to look at, designing experiments where we look at the response of plants to climate change, possibly with a view to select cultivars that can respond in a certain way to the climate change. But what I'm really interested in is capturing this variability and being able to look at the cultivar selection through the lens of the variability, of partitioning the variability. And I'm, of course, interested in a disease and pest response to climate change, again, being able to introduce different sources of variability in a controlled way. So just summarizing and explaining why the picture that I put when I was asked for a picture that characterizes the work, I didn't want to include highly stylized radish sun systems, but instead showing this field visit in India. And that's really the picture that to me underlies some of these problems, because when we're looking at the field here, I mean, there is this question that John posed how many years ago, which is why those plants are different. And to the farmers like this one, the variability and an associated risk of the outbreak or risk of the bad response of the crop to drought or other conditions is actually something that's important for their livelihood. So not every plant is the same. There is a genotypic, phenotypic variability. There's environmental variability. It's something that is difficult to explain, which we call the stochastic variability. And of course, there is one element in which those variabilities are important, and that's where pests and diseases are introduced into the system. And many agricultural systems, like the one that I'm showing here, are on the cusp. I mean, even a small change in a certain direction can cause the system to collapse. So I want to stress here, just going back to my first slide, which is that variability is clearly a problem. The variability for this farmer, I mean, this farmer would like to have crop a year after year or season after season, and would like to have as uniform yield as possible. So for him, the variability is a problem. But the variability, and particularly our understanding of variability, is a solution. But how can we understand the variability? Well, that requires a combination of experimentation and modeling analysis. And that experimentation, we need to start looking at the high replication and very fast and high throughput techniques. And on the modeling side, where we're moving into is utilization of modern technologies, for example, the use of artificial intelligence in assessing the architecture of plants and the, for example, leaf area. And also using advanced statistical methods from data science, looking at the Bayesian parameter estimation. And linking that to epidemiological modeling will give us an idea of how we can start to understand the variability. Thank you very much for your attention.
Thank you very much for a fascinating talk, Adam. And I think as the Advanced Plant Growth Centre comes on stream, some of the approaches that you're applying will be really useful, and perhaps you won't have to travel quite so far to get data that you can work with. So we should keep talking.
Yes,
so what I'd like to do at this point is just invite anybody in the audience to either type a question into the chat box, or alternatively feel free to unmute and just go ahead and ask a question directly. Okay, so no, I don't think Ian, please go ahead and feel free.
Adam, thanks for that, a great talk. When you were talking about example 2, you were talking about a situation where you had something that didn't show symptoms but would go on to cause a problem later.
Yeah.
But if it causes a problem later, I guess that means it has symptoms later. So how do you differentiate between no symptoms and something that doesn't have symptoms at that point but does have symptoms at that point?
Sure. Well, I mean, what we did, yes, that's in an experiment. So there are these two elements to this. I mean, first one is what happens in real life is that a tray is being transferred from the propagator to the field, and essentially, well, some plants might be showing symptoms later, or some plants might not be showing symptoms at all. but they will be colonized by the fungus, and the fungus can then start growing in a field. The other way in which that can be manifested is plants become stressed when being planted out into the field. So some of them might be not showing symptoms and might be perfectly okay, even if we were growing them longer in a glass house, but after being transferred into the field, they will develop symptoms. In this case, well, essentially, I'll just go to the model. We've assumed here that the plants go from susceptible into the pre-symptomatic stage, where they don't show symptoms, and then they move into the symptomatic stage. So the plants, as you pointed out, will move from one stage to another. So we're assuming that at some point, every plant will become symptomatic, which, as I said, might not be the case in real life.
Okay, thanks, Adam.
Okay, does anyone else wish to ask a question from the floor? Perhaps while people are pulling their thoughts together, Adam, I could ask a question. So in your example one, I think it was with one of your trichoderma species, you demonstrated that the biocontrol was effective. That's the viridae was effective against primary infections.
Yes.
But the Harzianum was effective against primary and secondary. So is that just an inoculum dose effect, do you think, or is everything understood about why the mechanisms may be different between primary and secondary infection?
And the answer is I don't know. We didn't study that, and this is actually, I mean, the results I'm showing here. I don't think we actually published the Harzianum work, and unfortunately, we won't be able to go back and study it. I'm afraid.
And would there be any way of perhaps modelling how inoculum size influences progression to disease?
Yes, I mean that could be. I mean, there is. We did some other work that I'm not showing here, where we looked at individual experiments, and when we challenged plants with inoculum and looked at how quickly they become infected. Yes, it could be. We could bring in the size of inoculum into this. Absolutely. But I mean, we would need to be guided by experiments. I mean, we just did this experiments with one size of inoculum.
Yeah. Okay, thank you. Does anybody else have any final questions for Adam? Clearly a very well presented talk, Adam, as all questions seem to be answered. So thank you very much again for joining us and such an interesting presentation. As I say, please do keep in touch with the Advanced Plant Growth Centre, and as things move on, I'm sure there'll be opportunities for us to work together. Yeah. So thank you again, Adam. Thank you.
Thank you. Thank you very much.